Hello, so today we're presenting our protein for the protein project, and our protein is called Sleepin, and it is created by Stephen and Sandy. So this slide shows the primary structure of our protein, and it consists of 53 amino acids. Over here, we created a table to let you guys know whether the amino acid is polar, nonpolar, acidic, etc. And from the photo, there are two polypeptides, the A polypeptide and the B polypeptide. The A polypeptide is noticeably longer than a B polypeptide. And for each N, we decided to label it the N-terminus and the C-terminus. This is because for the N-terminus, it contains an amino group, and for the C-terminus, it contains a carboxyl group. And since you guys can't really see the colors, we decided to create a model. And from the model, we can see that there are 19 polar groups, 14 nonpolar groups, 10 basic, and 10 acidic group. And this is the primary structure for our protein because it shows the sequence of amino acids linked together by peptide bonds. And peptide bonds are formed through dehydrogenase synthesis, where the OH from the carboxyl and an H from the amino interact to release a water molecule that forms the peptide bond. And because of peptide bond, we now have the primary structure for our protein. And this shows the secondary structure of our protein, and the secondary structure is essentially the folding of the polypeptide that results in an A helix or B pleated sheets. And we decided to represent our secondary structure as A helix. And the secondary structure is stabilized through hydrogen bonds. And over here, we show you guys the hydrogen bonds between histidine and aspartic acid. And over here, we show you guys the hydrogen bonds between glutamic acid and glutamine. And the hydrogen bond forms between the O from the carbonyl group and the H from the amino group. For the tertiary structure, we decided to show you guys the bond between cysteine and cysteine. When we have a cysteine and cysteine, they form a disulfide bridge. And since these two cysteines are from the same polypeptide, we decided to call it an intrachain disulfide bridge. And it is essentially a nonpolar covalent bond.